Hi everyone, this is Alex Antonio, the founder and CEO of Fins Attached, and I'm so excited to be a part of this event today here, and we're here to explain to you a little bit about what Fins Attached is all about, our mission, what we're doing, and introduce you to our research vessel, the Sharkwater. So we're at Cocos Island, Costa Rica right now, which is about 360 nautical miles off the Pacific coast of Costa Rica. And you can see my background here is this beautiful, lush island. This is one of the rainiest places on the planet. It gets about 350 inches of rain a year. So that's why it's so lush and green and vibrant here. So Fins Attached is a nonprofit. We're a 501c3 nonprofit organization out of Colorado Springs. And our mission is to conduct research, promote conservation and education, and advocate for the protection of our oceans. We were founded in 2010, and since then we've embarked on our mission to study sharks and collect the valuable data that is needed for the protection and management of these critically endangered animals. So our mission is to provide data for government, so it's data-driven conservation. So we study sharks, we tag sharks, we track their movements, we collect tissue samples for DNA and population dynamic research as well. Because once we know where these animals are going to and coming from, then we can have a better idea on how to manage the populations. And this is what the governments need, the governments like Costa Rica and Ecuador in this part of the world where we're currently operating. So that's what Fins Attached is all about. And in order for us to be able to go out into the field and conduct this valuable research, we have the vessel that I'm standing on right now. We're in the bridge of the incredible research vessel Sharkwater, which has an interesting history, and it's kind of ironic really, because this boat was originally built in 1997 in Japan and was used as a Japanese fishing boat. How about that for irony for you? So now instead of this vessel taken from the ocean, it's now given back to the ocean. So we've redeemed this boat. So you can see the instrumentation and the navigation computer and our radars and how we maneuver and our radio equipment over here. But this vessel allows us to do some incredible work, gets us to these remote locations. It has a range of like 7,500 nautical miles, incredible stable platform. It was built for the high seas. So this is what this vessel allows us to do. So every expedition that we go out, whether it's Cocos Island where we are now, or whether it's in Mexico or in Galapagos, it's all about doing the research that ultimately gives us the data to present to governments for their management programs. So this is an example of one of the cabins on board Sharkwater. This is the master cabin, the owner suite. Has a private bathroom. All cabins, guest cabins, donor cabins have private bathrooms and showers. And no one said we had to rough it when we're out here on these research expeditions. Here we are downstairs on Sharkwater, and I'm here standing next to this poster of this gentleman called Rob Stewart. Rob Stewart, about 15 years ago, launched a film called Sharkwater, and it had such a profound effect on people's psyche about sharks on a world basis, and people started thinking differently about sharks. And so Rob was filming for his second film, the sequel, Sharkwater 2, which ultimately got called Sharkwater Extinction. And he had a diving accident while he was filming for that sequel and tragically died. So in his honor, we're actually named the boat Sharkwater after his original film. And so people can become a sponsor. We have youth, adults, and corporate. And as you see here, we have the Premier Advisor Program. We thank you so much for your sponsorship and participation and endorsement about what we're doing. And so this is what this project is all about. So normally when we're out at these remote locations at Cocos Island, we're going out on dives during the day, and during the dives we try to tag sharks externally using a pole spear. We're also servicing receivers which have collected data from sharks that have been previously tagged. So right now we're actually getting ready to go out on our third dive of the day. So follow me, let's go down to the dive deck. So here we are now on the dive deck area, the prepping area of Sharkwater. And as you can see, the divers are now getting ready to go out on the third dive of the day. They have pole spears with tags or biopsy tips on the end of pole spears so that we can either tag or collect tissue samples from the animals. And then over here, this is the camera table area for prepping of the cameras and also of the research equipment. Over here we have our local chief scientist. This is Randall Arals, who's the International Policy Director for Fins Attached. So what are we doing on this dive, Randall?
Well, we're gonna try to tag some hammerheads or other species. We're also taking tissue samples. So it's all part of, the bigger picture, the bigger projects.
Okay, great. And so you have a couple of assistants going out with you, right? So we have at least 2 or 3 assistants going out with you who are going to help with the tagging and the tissue sampling. And then, so this is where people suit up. This is all the scuba equipment where they suit up for the diving. And then over here, this is Isak. Isak is one of the park rangers, one of the head park rangers here at Cocos Island National Park. So we work in partnership with Isak, Fins Attached, as well as the park services. We have a cooperative agreement, so it's a partnership collaboration to make sure that we're studying the sharks and getting the data that we need that these guys can use for their management. Say hi, Isak.
Hey, keep coming.
Audio Jungle. 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 Audio Jungle.
Okay. ¿Listos?
Uno, dos, buena hora. Tres.
So here we have a silky shark that we just pulled up. Silky sharks are a highly migratory animal, and they've been known to migrate for thousands of miles. So now what we're doing, the scientist here, Jeffrey, is doing an incision right between the pectoral fins to put a tag internally. And the internal tags are preferred to the external because these tags have a battery life of 10 years. And when we tag them externally, the tags fall off prematurely, so we don't maximize the amount of data that we get. So we do internal tagging so that the tag stays with the animal for the entire duration of the tag's battery life. So now he's putting the tag inside, and then once the tag's inside, he's going to suture up the incision. So now we're going to be measuring the animal. So we put the measuring tape right at the tip of the nose. The two main measurements that we took was the fork length from the tip of the nose to where the tail, the two caudal lobes of the tail meet, so the fork length, and then the total length, so from the tip of the nose to the tip of the upper lobe of the caudal fin or the tail fin. So those are the two main measurements. And then we take a tissue sample.
No, we already did.
So we take a little clip from the second dorsal fin as part of the tissue sample for DNA work.
3 minutes.
So you'll hear the person timing, so 3 minutes. So we want to minimize the stress on the animal by keeping it out of the water for as little time as possible. So when it comes to knowing the migration of these animals, it's important for governments to know where these animals go, how long they spend in different areas, because hopefully that becomes part of the management plan for that country. Or in cases of highly migratory, multiple countries, because it has to be international policy changes between countries if we're going to have any hope in protecting these animals. Now the last thing we do is we take the tail rope off, we take the hook off of the mouth, and then the shark goes off and swims off.
4 minutes.
You know, this looks like a pretty traumatic event for the shark, but these sharks are very robust animals. These silky sharks, these Galapagos sharks, they're very robust and they recover fairly quickly. And we saw that one swim off nicely once it got back into the water. But it's a necessary part of the work to collect the valuable data, because it's got to be data-driven conservation. That's what this is all about.
Audio Jungle.
So here we are back on the bridge of Sharkwater as we're heading back to the marina in Quepos after a very successful expedition to Cocos Island. We were able to tag 5 sharks internally off the back of the vessel, Sharkwater, but we were also able to tag 2 tiger sharks. The tiger shark is a species that we have a main focus on. And some of these animals, some of these sharks are critically endangered. And so we're always looking for help, and invariably people ask us the question, what can I do to help? What can I do back in my home, back in my town, back in my state to help? Well, there's always the simple answer of becoming a member of Fins Attached, making a donation, becoming a sponsor. But there's things beyond the money component that you can do to get involved. Sometimes we look for people to sign petitions. Sometimes we're looking for people to write to their congressman. Right now there's a bill going through the government in the US to ban the sale and possession of shark fins on a federal level. So writing to your congressman to support that bill would be a tremendous thing to do. So there's other things that you could do to get involved with Fins Attached. Go to our website at finsattached.org and you can learn more about what we're doing and our mission. And thank you for being a part of this expedition. Thank you for your support and your sponsorship, and we look forward to working with you in the future.